After the process of transcription has occurred in protein synthesis, which happens in the nucleus, the piece of mRNA that carries the code for the protein will end up leaving the nucleus through one of the pores, travel to the cytoplasm, and enter translation. So that mRNA will travel to the cytoplasm where it will attach to a ribosome like I've shown over there, but I've made the mRNA molecule slightly longer to be a bit more representative of actually how it would be in real life. So this structure here is the ribosome. We can tell that this is the ribosome because it has this upside down snowman-like structure, because ribosomes have a small part to it, which we call the small subunit, and a large part, which is the large subunit. So the ribosome consists of rRNA, ribosomal RNA, and ribosomal RNA actually helps to bond amino acids together according to the code on mRNA. So this structure here that is going through the ribosome is still the mRNA molecule, and the ribosome will read the codons on this mRNA molecule. So what the codons are is 3 bases that are in a row. So there's 1, 2, 3, that is one codon. 1, 2, 3, that is another codon. And that is how the mRNA molecule is read, in order of those 3 consecutive bases that make up a codon. So codons are represented by 3 letters in a row. So in this case, AUG is one codon, GAC is the second, and so on and so forth. But for simplicity's sake, we only have 4 codons here. This molecule will go on much longer because you need 50 codons, so 50 amino acids to make a protein. But for simplicity's sake, I'm just showing you 4 codons here, the first, second, third, and fourth, just to keep things a little bit simpler. So in this protein, there will only be 4 amino acids, so it's technically a polypeptide. So let's finish labeling this process just to point out the structures, and then we'll do the step-by-step of what happens. So this funny-looking molecule here that seems to have these sticky outy bits, this is what how we represent a tRNA molecule. So this whole structure is actually tRNA, which is a little bit weird. And at the end of this tRNA, on all of them is an anticodon. So that's an anticodon, that's an anticodon, that's an anticodon. So these anticodons are on tRNA, whereas mRNA has codons. So anticodon on tRNA. Now, tRNA molecules carry these things on the top here called amino acids. So each amino acid has a specific name. You do not need to learn it, but we'll give you a table and I'll show you how we can figure out the names of amino acids. So this, for example, is leucine, this is tyrosine. And tyrosine will always be carried by a tRNA that has UAC, or some other options as an anticodon. But if there is a UAC, it will always carry tyrosine as an amino acid. So that is actually our first step of translation. The fact that each tRNA carries a specific amino acid. That is going to be our step 1. Step 2 relates to the fact that the tRNA anticodon needs to match with the mRNA codon. So as the ribosome is reading the codons on mRNA, the first codon is AUG. So what would be complementary to the A would be a U on tRNA, complementary to the U would be an A, and complementary to the G would be a C. So then it is this tRNA molecule that will bring its required amino acid to the ribosome. As the ribosome reads the second codon as GAC, the complementary anticodon has to be CUG. So then that tRNA molecule is called to bring its amino acid to the ribosome. Once the tRNA has dropped off its amino acid that is required, that tRNA molecule will leave the ribosome and go back to the pool of amino acids that sit in the cytoplasm. So if they are required to bring another tyrosine or another leucine, then they can do so. Now these amino acids will grow in a chain, forming what we call peptide bonds. So that would be our next step in protein synthesis, that the amino acids become attached to one another by peptide bonds. So this process will happen many, many times throughout the entire mRNA molecule. Eventually, you will end up with a chain of 50 different amino acids. Some of them obviously will be repeated, But this chain of amino acids will be 50 amino acids long. And if you think about it, each amino acid was coded for by one codon, which consists of 3 nitrogenous bases. So for every amino acid, so here you have 4 amino acids, you will have 4 codons that coded for those amino acids. Then if you think about it, how many nitrogenous bases there are that code for these 4 amino acids, there will be 12 different nitrogenous bases in a row, or 12 nucleotides even, because one nucleotide has 1 nitrogenous base. So if you think of tyrosine having 3 nitrogenous bases or 3 nucleotides, 1 codon. Leucine, 1, 2, 3 nitrogenous bases, 1 codon. Methionine would have also been 1, 2, 3 nitrogenous bases, 1 codon. Alanine, 1, 2, 3 nitrogenous bases, and 1 codon. So it's important to understand the relationship between number of amino acids in a chain and number of codons. It's 1 to 1, because it's 1 amino acid for 1 codon, because there's 1 anticodon. Or between the number of amino acids and the number of nitrogenous bases or nucleotides. And then that is a 1 to 3 ratio. In the next video, I'll do a brief explanation of how to answer questions when you asked about these amino acid tables, where you have to know what anticodons code for each amino acid or codons. And these are very, very simple examples to score very good marks. But for the process of translation, you need to learn these 4 major steps. And they are very easy to score marks on these types of questions. So please make sure you learn them as they are stated in these steps in the video.